Welcome to the deep dive. Today we're taking a closer look, a statistical scalpel really, to one of the hottest tools in conservation, environmental DNA, or eDNA. We're moving past the fieldwork and lab benches, diving deep into the data itself. Because, well, according to a big new study, the way scientists are crunching the numbers might actually be undermining the whole field. We're focusing on this paper. From anarchy to clarity, data pre-processing and statistical choices influence quantitative environmental DNA, eDNA analyses. It's really important work, led by Bylemans and Everts, along with Brys and Richard P. Duncan.
Yeah, and our mission for you today is basically to cut through some of that complexity. We want you to really get why these statistical decisions matter so much. It's all about understanding how the choices researchers make after collecting the eDNA, how they clean it up or summarize it or which statistical tests they pick, can totally change the story they tell about species abundance or biomass. This paper really lays out a potential roadmap for getting some standardized best practice guidelines, which honestly the field seems to desperately need.
Right, because eDNA is fantastic. It's non-invasive, pretty cost-effective, great for just figuring out if a species is there. But when we try to go further, when we try to quantify, like estimate how many fish are actually in that lake based on the DNA floating around, that's where things get tricky. The goal seems simple enough. Find a solid link between the DNA copies you measure and the actual number of critters, right? So why the anarchy, as the paper puts it?
Well, it's because while the community's done a pretty good job standardizing how you collect the water samples or how you run the DNA tests in the lab,
Yeah.
there's this huge gap when it comes to the analysis. What happens once those raw numbers land in the spreadsheet? It's kind of a statistical free-for-all right now.
And that matters because, well, if everyone's using a different recipe to analyze the data, the results aren't just potentially shaky, they're impossible to compare or even reproduce, right?
Exactly. You lose that reliability.
Okay, so before we get into the questionable recipes people are using, why is this raw eDNA data so hard to handle statistically in the first place? What makes it so messy?
Yeah, the raw data itself has some inherent characteristics that just don't play nicely with basic statistics. There are really three big challenges. First off, you've got this hierarchical structure. Think about it. You might run, say, 5 PCR tests, technical replicates, on just one water sample. And maybe you took 3 water samples from one spot, one site. Those 5 results from the same sample. They're not independent, they're nested within that sample. Same for samples within a site. You can't just treat every number as separate.
Okay, so they're related, not independent measurements. Got it. What's next?
Second is over-dispersion. Now, eDNA data are counts, 100 DNA copies per liter, maybe 500 copies. Technically, counts like these should follow a nice predictable pattern called a Poisson distribution. But with eDNA, the actual spread, the variance, is way bigger than the average count. It's much clumpier and more unpredictable than a simple Poisson model expects.
Right, not a smooth curve. More like peaks and valleys.
Exactly. And finally, there's zero inflation. You just get way more zero counts than you'd expect statistically. A zero could mean the species really isn't there, but it could also just mean you failed to pick up the DNA, maybe the concentration was super low, or something went slightly wrong in the lab step. It's a false negative, potentially. Standard statistical models just aren't built to handle this huge heap of zeros properly alongside the other issues.
Okay, so we have data that's nested, clumpier than expected, and packed with potentially ambiguous zeros. Quite the statistical beast. So to figure out how researchers were actually dealing with this, or maybe not dealing with it, the authors did this huge literature review. They looked at quantitative studies trying to link eDNA concentration to actual abundance or biomass. And crucially, they tried to get their hands on the original raw data to reanalyze it themselves.
Yeah, and what they found about the current state of practice, well, it's a bit alarming, especially on the transparency front. They looked at 110 papers published between 2011 and 2023. The field's growing fast, that's clear. But the descriptions of the statistical methods used, often really vague. And here's the kicker for reproducibility. Out of 106 studies published up to 2022, only 26, that's less than a quarter, actually made their full, raw, untouched data available.
Wow, less than 25%. So you basically just have to take their word for it most of the time. You can't check the analysis.
Pretty much. And that lack of access makes it impossible to verify the claims or build upon the work reliably.
And this lack of transparency, it seems, feeds right into that anarchy they talk about. They documented some common, maybe questionable shortcuts researchers were taking with the data they did analyze. Like what?
Well, a big one was summarizing. They found about a third of studies, 36% per sample or 32% per site, were just averaging the eDNA concentrations before doing any
Just
statistics.
taking the mean and
Yeah.
losing all that variation within the sample or site.
Yep. Just averaging it out. And then on top of that, about 1 in 5 studies, roughly 19%, were using filtering. This means they were deliberately removing data points, usually the zero counts, or values below what they call the limit of detection or limit of quantification.
Okay, hold on. Why would they do that? Average or filter? Is it just to make the stats easier, or is there some logic, however flawed?
It's probably a mix of things. With filtering, the rationale is often that anything below that LOD threshold, the point where you can confidently say DNA is present, is just noise or maybe contamination, right? But the problem is, by throwing out those low values, especially the zeros, you're potentially removing real information. Information about true absences or about the variability at low concentrations. Good models actually need that information.
And averaging.
Averaging is often done to try and meet the assumptions of simpler statistical tests. Like, you force the messy, nested data to look like clean, independent data points so you can run a basic correlation. It simplifies things, but you lose crucial detail.
Okay, so they're simplifying the data before the analysis. What about the analysis itself? Were people using the complex models needed for this kind of data?
Mostly no, it seems not. While some used more appropriate generalized linear models, which was the most popular category at 53%, a significant chunk still relied on really basic methods like Spearman correlations, that was 15%, or Pearson correlations, another 7%. These simple correlations basically assume your data is nicely distributed and all points are independent, which, as we just established, eDNA data definitely is not.
Right, so using the wrong tools for the job,
Very
essentially.
much.
Okay, so this leads us to the really novel part of the paper, the reanalysis. The authors managed to get hold of 11 of those raw data sets, and they systematically tested different approaches. They applied those common pre-processing steps like averaging and filtering, and then ran different statistical models on the results, basically stress testing the common analysis recipes.
Exactly. They wanted to see how these choices actually impact the results specifically. The ability to detect those significant positive correlations between eDNA and abundance biomass and the strength of that correlation.
And this is where it gets really, really interesting. When they compared the different statistical models on the raw, unprocessed data, they found overwhelmingly that one type of model consistently fit the best. The optimal choice for raw quantitative eDNA data. It's the negative binomial mixed effect model, or NB model.
That's the headline finding, really. The negative binomial distribution itself just handles that clumpy, overdispersed count data much better than simpler options like Poisson or even log normal distributions. And the mixed effect part accounts for that hierarchical nested structure we talked about, the replicates within samples within sites.
Okay, so the NB mixed model is the best fit. What happened when they used those other simpler tests, like the Spearman and Pearson correlations, on the raw data?
Well, that led to a significant increase in type I errors.
Type I error. That's a false positive, right? You think you see a significant link between eDNA and abundance, but it's not actually real. It's a statistical fluke.
Precisely. You're finding phantom correlations. So using those simple, inappropriate tests inflates your chances of claiming a discovery that isn't actually there.
Okay, so that's the impact of choosing the wrong test. What about the impact of that pre-processing, the averaging and filtering?
Yes. That had dramatic effects too. When they mimicked what researchers were doing by averaging data, taking those means per sample or per site before analysis, they saw a major overestimation of significant positive correlations. And the estimated strength of the relationship, the slope, was also inflated.
So averaging makes it look like there's a stronger relationship than there really is. It creates false confidence again.
Exactly. It smooths out all the natural noise and variability, making weak or non-existent correlations appear statistically significant. This was particularly bad when averaging was combined with a suboptimal model, like a basic Poisson model.
Wow. So averaging is like photoshopping your data to look cleaner, leading to misleading results.
That's a great way to put it. You're essentially modeling a simplified artificial version of reality, not the messy truth.
And what about the opposite approach, filtering out the zeros?
Filtering had the reverse effect. When they removed the zero values before analysis, it actually led to a decreased ability to detect true positive correlations. So you end up with an increase in type 2 errors, false negatives. You miss relationships that are actually there, and you underestimate the strength of the correlation when you do find one.
Unbelievable. So, depending on how you massage the data beforehand, you could either invent a correlation that doesn't exist or completely miss one that does.
That's the core message. The choice of pre-processing strategy and statistical test isn't trivial. It can fundamentally change your conclusion, swinging you between false positives and false negatives.
The implications are huge. This really underscores the need for harmonizing these analytical workflows. QDNA's credibility kind of hangs in the balance.
Yeah, and think about the real-world impact. Conservation managers, policymakers, they rely on these studies to make decisions about funding, about protecting species, about regulations. If, say, a third of studies are using averaging and potentially inflating false positives, how much effort and money might be misdirected based on correlations that aren't robust?
Or the flip side, if filtering leads to false negatives, maybe we incorrectly conclude an endangered species isn't responding to habitat restoration because its faint eDNA signal got filtered out as noise.
It undermines the trust in eDNA as a quantitative tool if the results are so sensitive to these hidden analytical choices.
Okay, so to combat this anarchy and bring clarity, the authors propose some clear recommendations. Let's walk through them. What's the first step?
The absolute foundation, number one, is increased transparency and data availability. This is just non-negotiable for science. Researchers have to provide complete, detailed descriptions of their statistical methods. And critically, they need to make the raw, completely unmodified data publicly available, along with good metadata explaining it.
That less than a quarter statistic for data sharing really needs to change then. It has to become the norm, not the exception.
Absolutely.
Okay, what's recommendation number 2? The preferred method.
Number two is about the preferred statistical model. Based on their reanalysis, the strong recommendation is to use negative binomial mixed effect models for assessing these correlations. These models are equipped to handle all three challenges simultaneously: the hierarchical data structure, the overdispersion in counts, and the excess zeros. They simply provide the best, most statistically sound fit for raw eDNA count data.
And they mentioned a backup.
Yeah, they acknowledge that fitting NB mixed models can sometimes be tricky, so they suggest log normal models could be a secondary alternative, but only if you absolutely preserve the raw data structure, including the zeros, perhaps by adding a small constant before log transforming. But NB is definitely preferred.
Got it. And the final key recommendation.
Number three is basically avoid pre-processing. Those common strategies like summarizing data by taking means or filtering out zeros or values below the all-audio queue, just don't do them. Especially don't combine them with those suboptimal statistical tests like basic Poisson, Spearman, or Pearson correlations. That's where the errors really got amplified in their analysis.
So embrace the messiness of the raw data. Don't try to artificially clean it up before analysis.
Exactly. And that's why the NB mixed effect model is so powerful. It doesn't require you to simplify the data first. It's designed to model the complexity directly: the counts, the extra zeros, the nested structure, all of it. It respects the variability that comes from the real biological and sampling processes rather than ignoring it like averaging does. That leads to much more reliable conclusions.
So this deep dive into Bylemans et al. Really hammers home that good science doesn't stop at the lab bench. Methodological rigor has to extend all the way through the statistical analysis. The path from anarchy to clarity seems paved with negative binomial mixed effect models and open data.
Yeah, though we should briefly mention the scope. Their analysis focused mainly on freshwater fish studies using specific quantitative methods like qPCR and dPCR. They deliberately didn't include studies reporting results as like nanograms per liter, because modeling those kinds of zero-inflated positive decimal values brings its own set of statistical headaches they didn't tackle here.
Right, good point. So what's the big takeaway for you, the listener? I think it's that the integrity of this powerful eDNA technology hinges just as much on the statistical choices made after data collection as it does on the sampling and lab work. Understanding that these choices drastically affect the outcome means we need standardization and transparency in the analysis just as much as anywhere else in the process.
Absolutely.
Which leaves us with a final thought to mull over. Given this strong evidence that very common practices like averaging data significantly hike the risk of false positives, what percentage of the existing quantitative eDNA literature, the studies already published using those methods, might need a second look? or at least need to be interpreted with a hefty dose of caution before we bet conservation resources on their findings. Something to think about as the field works towards that clarity.